Bioanalytical GLP consultants are experts who guide organizations in complying with good laboratory practice, GLP, regulations for bioanalytical studies. They play a crucial role in ensuring that studies meet regulatory standards for accuracy, reliability, and reproducibility. This presentation will explore the world of bioanalytical GLP consultants, regulations, and their impact on the pharmaceutical and biotechnology industries. GLP compliance consulting involves advising on the setup of GLP-compliant bioanalytical laboratories and guiding best practices for handling, processing, and analyzing biological samples. Regulatory support includes assisting in preparing study protocols and final reports that meet regulatory expectations, ensuring compliance with OECD GLP and 21 CFR Part 58. Training and development offers training sessions for laboratory personnel on GLP principles, including correctly handling and reporting bioanalytical data. Method validation ensures that bioanalytical methods used for testing are validated under GLP standards, meeting accuracy, precision, sensitivity, and reproducibility requirements. Quality assurance in bioanalytical GLP involves implementing QA systems, performing audits, maintaining documentation, and continuous improvement. Establishing internal quality assurance systems ensures continuous GLP compliance. Regular audits are conducted to ensure bioanalytical studies meet all GLP standards. Proper documentation and record-keeping are essential for all QA activities. Continuous improvement identifies areas for enhancement and implements corrective actions. Bioanalytical GLP regulations ensure that studies analyzing biological samples for drugs, metabolites, or other substances produce reliable, reproducible, and credible results for regulatory decision-making. Key components include method validation, sample handling and storage, instrument calibration, data integrity and documentation, quality control, and reporting. Method validation in bioanalytical GLP ensures specificity, accuracy and precision, linearity and sensitivity, and stability. Specificity ensures the method can distinguish the analyte from other components in the sample. Accuracy and precision verify the closeness of test results to the true value and the consistency of repeated measurements. Linearity and sensitivity confirm the method's ability to produce results proportional to analyte concentration and detect low levels of the analyte. Stability assesses the analyte's stability under various conditions and storage times. Sample handling and storage in bioanalytical GLP involve collection, labeling, storage, and monitoring. Proper collection techniques ensure sample integrity and prevent contamination. Clear and accurate labeling of samples prevents mix-ups and ensures traceability. Appropriate storage conditions, including temperature and humidity, maintain sample stability. Regular monitoring and documentation of storage conditions are essential. Instrument calibration in bioanalytical GLP involves regular calibration, maintaining calibration records, verification, and documentation. Ensuring instruments like chromatographs and spectrometers are calibrated regularly is crucial. Maintaining detailed records of all calibration activities is necessary. Verification involves checking calibration accuracy through standard samples and controls. Properly documenting all calibration procedures and results is essential for easy access during inspections. Data integrity and documentation in bioanalytical GLP involve data security, comprehensive documentation, secure archiving, and audit trails. Implementing robust systems to protect data from unauthorized access or manipulation is crucial. Maintaining detailed records of all raw data, calculations, and analyses is necessary. Storing data securely enables future access for review or audits. Implementing systems to track all changes made to data, including who made the changes and when, is essential. Quality control in bioanalytical GLP involves internal quality control measures, use of controls and standards, system suitability tests, and continuous monitoring. Implementing measures to ensure consistent performance of analytical methods and instruments is crucial. Using controls and standards during analysis verifies method accuracy. Conducting tests before each run ensures the analytical system functions properly. Regularly reviewing quality control data identifies and addresses any issues promptly. Reporting in bioanalytical GLP involves compiling comprehensive reports, ensuring GLP compliance, clear communication, and timely submission. Detailed reports include raw data, statistical analysis, and any protocol deviations. Ensuring reports adhere to GLP guidelines for completeness and accuracy is crucial. Presenting results clearly and understandably supports regulatory review. Submitting reports within required timeframes supports regulatory decision-making. Applications of bioanalytical GLP include preclinical studies, method development, clinical studies, toxicology studies, and regulatory submissions. Preclinical studies determine the pharmacokinetics and metabolism of new drug candidates. Method development involves establishing and validating robust bioanalytical methods. Clinical studies monitor drug concentrations in biological matrices during human trials. Toxicology studies analyze toxic substances or metabolites in biological samples. Regulatory submissions support IND applications and NDAs with GLP-compliant data. The OECD GLP guidelines provide a framework for ensuring that non-clinical studies, including bioanalytical studies, are conducted consistently, reliably, and reproducibly. They aim to generate trustworthy data for regulatory decision-making. Key components include organization and personnel, test systems, study plan, performance of the study, data integrity and documentation, quality assurance, final report, and archiving. Organization and personnel in OECD GLP involve establishing clear roles and responsibilities for all personnel involved in the study. A qualified study director is appointed, responsible for overall study conduct. An independent QA unit is established to ensure GLP compliance. All personnel are adequately trained and qualified for their roles. Test systems in OECD GLP involve appropriate selection, ethical considerations, proper handling, and documentation. Choosing test systems suitable for the study objectives is crucial. Ensuring compliance with ethical standards, particularly for animal studies, is necessary. Implementing procedures for proper handling and maintenance of test systems is essential. Maintaining detailed records of test system selection, handling, and maintenance is required. The study plan and performance in OECD GLP involve a detailed written plan outlining study objectives, clear methodology and protocols, defined data analysis methods, and documentation of any changes. Study performance includes adherence to approved protocols, consistent data recording, periodic monitoring, and documentation of deviations. Quality assurance in OECD GLP involves establishing an independent QA unit, conducting regular inspections, preparing compliance reports, and performing data audits. The independent QA unit oversees GLP compliance. Regular inspections ensure study processes and facilities meet GLP standards. Compliance reports are prepared for management and study directors. Data audits review raw data and reports for accuracy and completeness. The final report and archiving in OECD GLP involve a comprehensive summary of methodology, clear presentation of findings, detailed conclusions, and documentation of any deviations. Archiving includes secure storage of all records, preservation of raw data and reports, maintenance of study protocols, and long-term accessibility for review. (21 CFR Part 58) governs non-clinical laboratory studies supporting FDA-regulated products. It ensures studies generate data for regulatory submissions adhering to GLP standards. The FDA enforces these regulations, which are critical for drug development and regulatory approval processes. Key components of 21 CFR Part 58 include organization and personnel, study protocol, test and control substances, and quality assurance. Organization and personnel define roles and responsibilities, including the study director and specialized QA roles. The study protocol requires a detailed written protocol defining study objectives, design, methods, and analysis. Test and control substances mandate proper identification and handling. Quality assurance requires an independent QA unit to oversee study compliance with GLP standards. Performance of the study under 21 CFR Part 58 involves protocol adherence, accurate data recording, deviation reporting, and continuous monitoring. Ensuring studies are carried out according to the approved protocol is crucial. Implementing systems for consistent and accurate data recording is necessary. Promptly documenting and reporting any deviations from the protocol is essential. Regular oversight ensures ongoing compliance with GLP standards. Maintaining comprehensive records is crucial for all study-related activities and data. Long-term storage ensures that all data and reports are retained for at least 2 years after the final report submission. Secure access involves implementing systems to ensure secure storage and controlled access to archived materials. Audit readiness is about organizing archives to facilitate easy retrieval during audits or inspections. OECD GLP provides an international framework and guidelines for harmonizing GLP practices, offering more general frameworks with flexibility and is voluntary in some countries. In contrast, 21 CFR Part 58 is specific to the United States, mandatory for FDA-regulated products, and includes more specific and detailed requirements enforced by the FDA. Our consultants offer on-site GLP implementation, developing compliance systems, documentation, and workflows tailored to each organization. We provide comprehensive training on GLP requirements, bioanalytical testing, and data integrity practices. Quality assurance support assists in establishing and maintaining robust QA systems for ongoing GLP compliance. We also help prepare for regulatory audits and inspections to ensure GLP standards are met. Our expertise includes in-depth knowledge of GLP regulations and industry best practices. We offer continuous support to maintain GLP compliance. Customization involves tailored solutions to meet specific organizational needs. Efficiency is achieved by streamlining GLP implementation and compliance processes. Risk mitigation focuses on reducing the risk of non-compliance and regulatory issues.